Good morning, students. Today we'll discuss post-translational modifications in proteins. So let me start the presentation. So we'll discuss proteins and the post-translational modifications in proteins. So this is an interesting discussion today, and we'll see that how actually the proteins and their activities can be regulated, and proteins are the major building blocks in any living organism, and we'll see how through post-translational modification these proteins and their activities can actually be regulated. This is a very interesting chapter. There are a lot of biological stuffs involved in that, and finally we'll also see some of the computational methods, how we can actually predict post-translational modifications in protein sequences. So reviewing the basics, if we remember that when we had the DNA and in the genetic sequence, we have an open reading frame, and from that we try to identify the genetic sequence, and we have the open reading frame, and through the transcription process we have the messenger RNA, and this messenger RNA goes out of the nucleus and binds with a ribosome, which is also called a protein factory, to generate the protein. So this is called the process of translation. So first we have the transcription, and then we have the translation, and the nascent or precursor proteins are generated. So till now, even when we were discussing protein secondary structures and everything, we were doing things up to that level, and we roughly discussed that how a protein complex can be formed, but just output of a ribosome or the translation process is not just enough, and most of the actual inside cell biological activities and the regulations are often taken place after this step, and that we call as a protein processing and post-translational modification. And then we get actually the mature protein, and then based on the post-translational modifications and the processing, we have the proper folding and the biologically active protein is formed. So there is actually a huge difference from the encoding region that we try to see in an ORF, and then what we actually get as a final product as an active protein. And there are numerous variations, like one single gene can map to hundreds of different variations in proteins, and that is not static and just one to n mapping process. So there are many different dynamic variations that take place based on many different situations that in a specific cell. So we can say that post-translational modifications are numerous and diverse, and people are really working exhaustively in post-translational modifications. And to be honest, this is really a very advanced topic, and post-translational modifications are widely studied even today for many different biological activities and to understand many different biological processes that we continuously have within a cell or in any living organism, as a matter of fact. And that's for one of the reasons, like PTMs are called mediators of protein dynamics. As you can say that proteins are the main building blocks of a living organism, and PTMs are actually the mediators of these protein dynamics. They control, they have like the signaling pathways, they help in protein degradation, and like everything are actually modulated or mediated through different types of PTMs. So that is the major purpose of discussion today, and we'll see that if we again start with the messenger RNA, and during the transcription process, you will see that when in eukaryotic cells, you have 6 to 8 proteins for one gene based on different types of splicing, like how introns and exons are handled through the splicing process. So based on that, we have many different variations of proteins. So you can say that one single gene may not have a single messenger RNA that gets encoded through that gets decoded by the ribosome. So there is a 6 to 8 different variations for each gene that we have in our chromosome. And after the translation process, when we have the secondary the sequence information encoded from those messenger RNAs, we have many different variations of post-translational modifications. For example, 20 different forms of antitrypsin observed in human plasma. How can you answer that? So that can only be answered by the post-translational modifications in proteins. So we will see how these things are taking place. And what is it? So it is basically addition of groups or deletion of parts to make a finished protein. So you have a raw product, and then you actually need to finish it, and you need to add something extra, or you may crop somewhere. Only then it becomes a finished product, just very much like your diamond. And when you have your diamond, uncut diamond, so this is just like a dull stuff, so you need to cut it, polish it, so that you get a bright and diamond that actually people like. So what are the groups? So you have the methyl groups, acetyl groups, glyco groups, and the phospho. And in many places, in most cases, you have those side chains like the amino acid side chains, or the in the N terminus or in the C terminus, you have these post-translational modifications affected. And there are many different varieties, and there are many different purposes. So we'll definitely not cover everything, but we'll try to see some variations. For example, so you have the hydroxylations when an OH group is added, maybe a disulfide bond, a glycosylation, methylation, and lipidation. You have a protein, and then you have a ubiquitin added to it, then you get a ubiquitination. And so that is the post-translational modification, because you have a finished protein as this is your finished protein. And then once you have a ubiquitin attached to it, we call it a ubiquitination. So what is this? For example, ubiquitination is called the kiss of death process of a protein. What is this? In ubiquitination, a protein is inactivated by attaching ubiquitin molecule to it. Okay, so This is the post-translational modification process of a protein, which is called ubiquitination. So ubiquitin is a small molecule. This acts as a tag that signals the protein transport machinery to ferry the protein degradation. When means what is a proteasome? Proteasomes are protein complexes which degrade unneeded or damaged proteins by proteolysis. We'll see some of these examples even in our course and even today's lecture, that what is a proteolysis? A proteolysis is a process when it breaks the peptide bonds. Okay, so the enzymes that help such reactions are called proteases. So our point of discussion at this moment is post-translational modification. What are the different types of post-translational modifications? We can go and discuss every single type of post-translational modifications, and that is a huge, big research topic. And this is not the scope of that is not within the scope of this discussion, but what we are trying to stress here is what is post-translational modification. So we have a final protein, and then if we have a additional small molecule attached to it, or maybe it forms a diffusible bond, or maybe something else like a lipid chain is attached to it, then that same protein behaves in a different way. And that process of addition or deletion of something is called the post-translational modification. When you have the translation, you have the final protein. So post-translation, there are several modifications within the protein, and that actually mediates the protein dynamics. So that is absolutely important, and here we are showing one example of addition of ubiquitin, which is a small molecule that helps in protein breakage. When a protein is not utilized anymore, or it is not required anymore, then ubiquitin is attached to it in the post-translational modification called ubiquitination. And then we have the degradation. So that is called protein degradation. So there was a huge human proteome initiative. For example, we have around 21,000 human protein encoding genes, and you can see that through transcriptome we have close to 100,000 different human transcripts, that is the RNA transcripts. Okay, so from 21,000 we have close to 1:5 ratio, and we have many different splicing variants in our transcriptome. And even if we have only around 21,000 human genome, in human genome only around 21,000 protein encoding genes, we have close to 100,000 human transcripts, and that transfers to close to 1 million human proteins. 1 million, that is again 1:10 from each transcript. Like for one single messenger RNA can generate on an average up to 10 different types of proteins through different types of post-translational modifications, and they all act differently based on what additional molecule is attached to it, or how it is cleaved or broken to different parts, and that way they actually act as mature proteins. And these are, as we have seen, so this is mainly generated by alternate splicing and post-translational modification. So you can see that only from around 20,000, 21,000 21,000 human protein encoding genes, you have close to 1 million different types of human protein. So that is very hard to have a 1:1 correspondence from a gene to a protein. So till now we were actually discussing that we have a very simplistic scenario in all our previous lectures, that you have a genetic sequence, a nucleotide, and then you have a 3-letter codon that encodes a amino acid, and through the translation process you generate a protein, and you have a primary sequence, and then based on certain energy minimization and different types of bonds that takes place, and you have a secondary structure of the protein, then they fold, and tertiary structure of the proteins are formed, then you have the quaternary structure of the proteins are formed. So this is a very simplistic overview of the whole biological process that in a living cell. But in reality, we have these splicing variants and post-translational variants. So 20,000 genes encode 1 million proteins. So that is absolutely stunning, and we'll see how it works out. So coming into more detailed discussions, like what purpose, why, and what these post-translational modifications do. So what are their functions? Okay, so there are many different functionalities, and we'll just try to brief these functionalities with 1 or 2 examples here and there. So for example, for targeting. Okay, so PTMs are used for targeting, for example, in case of lipoproteins or lipoproteins. What is a lipoprotein? A lipoprotein is a biochemical assembly which whose primary purpose is to transport hydrophobic lipids, that is fats. Okay, so a lipoprotein is a biochemical assembly, or these are proteins which transport fat, okay, in water, as in blood plasma and other extracellular fluids. So it transports lipoproteins, they transport fat. Like, they transport fat to where? So maybe in muscles, where they are burned to generate energy. So fats are like small energy molecules. Okay, so they need to be transported. So they bind with the lipoproteins. The fat molecules, they bind with the proteins which are lipoproteins. So post-translational modifications often target lipoproteins so that can bind a fat. So that is a process, or that is one purpose, that is called targeting. So this is done through certain types of post-translational modifications. Post-translational modifications are also useful in case of stability. Different biological processes to enforce stability, there are different types of post-translational modification. For example, secreted glycoproteins. What do you mean by glycoproteins? Glycoproteins are proteins which contain carbohydrate chains covalently attached to amino acid side chains. So they are they have carbohydrate chains. Okay, and the carbohydrate binding process is known as glycosylation PTM. So there is a process like the glycoproteins which bind to carbohydrates, like the lipoproteins, they bind to fat. Glycoproteins bind to carbohydrates, okay, in their amino acid side chains. Okay, and the secreted extracellular proteins are often glycosylated, and they help in many different activities, for example, like including our immunity to help the immune system. So the glycoproteins are actually glycosylated, and they they are attached with the carbohydrate chains. There are many different variations. Okay, so in general, so this is also a purpose of like that helps in stability of a living organism. For example, if enough secreted glycoproteins are not glycosylated, then your immunity means our immunity system may fall down. Okay, so this helps in stability. Okay, there are often some specific functionalities of like for example, many viruses have glycoproteins that help them enter body cells. So to achieve a specific function, okay, so surface glycoproteins are again post-translatory modified in the post-translational phase to achieve a specific function. Okay, even in there are several virus proteins, they are modified to achieve a specific function. If we can, in some cases, if we can block that particular type of PTM, then probably that particular function can be inhibited. So different types of PTMs can help to achieve a specific type of function. So like here in case of viruses, maybe some surface glycoproteins help in anchoring, okay, and enter into the bodily cells. For example, we have seen that in case of COVID also, we have the anchors for this SARS-CoV-2, and they anchor with our certain proteins, and they enter into the body cells. So these type of functions can also be mediated, are also mediated through post-translational modification. Again, like control of activities, like the regulation. So, for example, the clotting and coagulation factors are heavily mediated through post-translational modification. So, what is coagulation factor? Coagulation factors are proteins circulating in our blood that are essential for proper blood clot formation. Okay, so that forms a gel-like formation when a blood clots. Okay, so those coagulation factors are different types of proteins that need to be properly modified so that a blood clot can be formed. So these are the general purposes of post-translational modification, and we'll discuss in more details throughout this discussion. I have some visualization of what we have just now discussed. For example, a plasma membrane protein can be linked to a membrane by a CGI anchor. So by a GPI anchor. So what is a GPI anchor? We'll discuss a bit more details, and we'll come to that again. But at this moment, we'll discuss that plasma membrane proteins can be linked. So this is a purpose of PTM. Various modifications regulate microtubular functions, target proteins like polyubiquitination can induce protein degradation, so that we have just discussed here. So this is the target protein, and you can see that ubiquitin is attached to that, and this is the proteasome, and then you can see that proteins are essentially degraded. So this process of attaching ubiquitin, so this is called polyubiquitination, that multiple ubiquitins are associated, and the target protein is degraded through this process. Likewise, like nuclear and cytoplasmic proteins can carry O-glycans. O-glycans are, these are basically carbohydrates. Glycans are essentially carbohydrates. Like the histone code control many nuclear processes. So histones are where our in our DNA, those nucleotide chains are actually bound around these histone cells. So these histone proteins. Okay, so these histone code control many nuclear processes. So these are also like methylated, ubiquitated, and there are a lot of different types of post-translational modifications, phosphorylated inside the nucleus as well. And this also helps in different types of regulatory activities within this. Plasma membrane proteins can carry these are like plasma membrane proteins. This can carry N-glycans. These nuclear and cytoplasmic proteins can carry O-glycans. They can carry N-glycans. Phosphorylation cascades are involved in many signaling pathways. So phosphate group is attached. So phosphorylation can also help in many different types of signaling pathways, like how a protein acts in a specific way. In general, they will not be active. When you add a phosphate or maybe a ubiquitin or some methylated, then it behaves in a specific way. So only when you do a specific type of, or only when a specific type of post-translational modification is applied, then only the protein behaves in a specific way. That is the purpose of this discussion. So this is just pictorial representation of what we were discussing so far. For example, you have all of us are familiar with insulin. Insulin is a very important protein that helps in our immunosystem. Okay, but the messenger RNA that encodes insulin is not exactly the insulin that our body is actually using. So it instead generates pre-pro insulin. Okay, and after specific types of post-translational modification, you can see that there are some cutting processes. Okay, so you have this first cutting process here, and we have this pro-insulin generated from here. Again, you have this, this is called protein cleavage, that the peptide cleavage, that these peptide bonds are actually broken between arginine and lysine, and you have you break out from this, and then it forms a active insulin. So until you have actually these successive post-translational modifications taken place, even if we have enough pre-pro insulin in our body, it will not act as a proper, it will not help our immunosystem. So these post-translational modifications are extremely important in that respect. So why post-translational modifications are made? We have already discussed some of them, and we'll discuss more the biological perspective and with some certain examples. But to be honest, like some of these examples are just for information purpose, because if you need to have a more detailed study, you can definitely go into them. And there are continuous research insights, like deep research insights against each example. So some of these biological examples are just given as simple terminologies, and we will not go into the deeper picture of this. Okay, so that is not within the scope of our study. So we will see, like in general, why protein post-translational modifications are made. For example, regulation. Regulation is a very important activity. So it regulates many different activities of protein, and there are interconvertible modifications. So sometimes proteins are modified and then they are converted back. Okay, so there are many. For example, you have a degradation process. Is it a unidirectional process or a bidirectional process? You can say that when a protein, through some post-translational modification, it degrades, like the peptide bonds are broken. So can they be again joined together? So these are points of discussion. So for example, sometimes we have regulation, and different types of regulations are there. For example, interconvertible modification. What do you mean by interconvertible modification? For example, there are some interconvertible enzymes, like there exist at least two well-defined reversible convertible forms produced by covalent modifications of amino acid side chains. Like one enzyme, after a specific type of post-translational modification, it takes another enzyme form, and that is a stable form, and then again you have some post-translational modification on that enzyme, and then again it comes back to the original enzyme. So these are called interconvertible enzymes. Like any enzyme that exists in at least two well-defined reversible convertible forms, often by covalent modifications of amino acid side chains. So most of the activities happen around those side chains that you have already studied for 20 different amino acids. Now, there can be many different types of interconvertible modifications, for example, using monocyclic cascade, some using cyclic cascade, like A affects B, B affects C, C affects D, and then again D from D to A. So there can also be unidirectional cascade. For example, proteolytic activation that I just gave example. Like you have activation of an enzyme by peptide cleavage. So you have a peptide bond and it breaks into several separate peptides, and this is an unidirectional cascade. So you cannot apply another post-translational modification and get back the original peptide. Okay, so these are the different types of regulations that take place. There are also cross-links. So another type is like cross-linking. What is cross-linking? Cross-linking is the process of chemically joining two or more molecules by a covalent bond. Chemically joining two or more molecules using a covalent bond. Attachment different groups of two different proteins result in intermolecular crosslinks and stabilize a protein-protein interaction. So, as you can see that we form protein complexes. So, these PTMs, post-translational modifications, they help in generating crosslinks so that either within the same protein they can form some kind of specific structures, like for example, like maybe in cysteine sulfide one can link together not just monomer but entire lengths of proteins to form secondary protein structure like beta sheets. So it can also form some modifications can also initiate change in the structure. Maybe it forms a beta sheet, or it can also help in stabilizing protein-protein interactions, where two different side chains are joined together through a covalent bond. So that is called a cross-link. So PTMs also initiate different types of crosslinks. Very common are diffusible bond crosslinks like cysteine-cysteine crosslinks. Okay, isopeptide where lysine is connected, transmutation, so that helps in blood clotting factor, coagulation, then tissue glutamase reaction, cell proliferation, aging, and all these things, programmed cell death. So that you can see that different types of actually amino acids, they can actually form different types of crosslinks, and that is done through different types of post-translational modifications. Again, I'm telling you that this is only for additional study, and we need to focus on the basic purpose, that is like in the previous slide you have seen that PTMs can wipe PTMs, so they are good for regulation. We have seen what are the different types, but are sample regulation types. They are good for they help in forming crosslinks. So we have seen, you know, like what are crosslinks and how they are done, but these are only sample examples. Okay, there can be many different varieties. You can have covalent cofactors. So cofactors are also very interesting stuff. So what is a cofactor? A cofactor is a non-protein chemical compound that tightly and loosely binds with an enzyme or other protein molecules. So we are not just talking about binding with one protein with another, or breaking a protein like peptide bond cleavage, or forming a peptide bond. There can be other non-protein chemical compounds as well, which bind tightly with a protein. Okay, so these are called cofactors. So PTMs can also help in binding cofactors in using covalent bonds. So cofactors are non-protein chemical compound that tightly and loosely binds with an enzyme or other protein molecules. A large number of these cofactors covalently link with polypeptide chain. So these are some of the examples of covalent cofactors, and there are many actually, and these have many different activities, especially in drug regulation and in disease pathway or disease diagnosis, and there are many different activities, like some PTMs allow some chemical compounds to bind in a specific way, and that they have very serious implications in disease propagation. So these covalent cofactors are extremely important in the disease analysis. Then there are membrane anchors. So we discussed briefly that there are different types of membrane anchors. What do you mean by membrane anchors? I'm just repeating. Many cell surface proteins are tethered to the extracellular membrane via GPI anchoring. That is one of the PTMs. So what do you mean by GPI anchors? They represent a post-translational modification of a protein with a glycolipid, and is used widely used in ubiquitously in complex organisms. So they help in binding with many different types of proteins, and these are called like membrane anchors. So these types of membrane anchors are enabled or disabled, or in a generic sense they are regulated with the different types of post-translational modifications. Okay, so these are some of the examples. Apart from that, there are many other different types of purposes like signaling, recognition, and structural amplification, and protein turnover like protein degradation, like ubiquitination, and spontaneous oxidation, and others like iodination, sulfation, or methylation. So there are many different activities that are regulated or mediated through post-translational modification. Now, what are the different types of post-translational modification? There are many different types of post-translational modifications. For example, post-translational modifications involving peptide bond cleavage. What do you mean by peptide bond cleavage? A peptide bond is there are different amino acids, and during the translation process, they form a peptide bond. An amino acid is also called as a peptide. They bind with each other through a peptide bond. Okay, and you can see, if you can say that there is a C terminus and an N terminus in any amino acid. Okay, so you can say that the N terminus is binding with the C terminus, forming a peptide bond. We have a small diagram here, so you can see that this is a single amino acid. So this is the alpha carbon, this is the C terminus, this is the N terminus. Okay, and what happens? This red one is the peptide bond. So you have this R1 is the side chain, and this side chain determines the type of this particular peptide or this particular amino acid, and this side chain determines the nature of this particular amino acid, and they are connected together through this peptide bond, and this is the C terminus and this is the N terminus. Now, using peptide bond cleavage, for example, in case of proteolysis, you have a water molecule that dissolves this peptide bond through a peptide bond cleavage, and they are separated. For example, in case of protein degradation, this peptide bond cleavages, they degrade a protein into smaller again amino acid. They become individual amino acids. Okay. Now, their peptide bond cleavage is a very important activity in post-translational modification. This is called limited proteolysis, where exactly we want that cleavage and how it has to be affected. That is very important. You remember, in case of pre-pro insulin, we have to have a cleavage at a specific position, and even in case of pro insulin, we need to have a cleavage in a specific position. If the cleavage is not in the desired position, we will not get the desired insulin. So specific and well regulated peptide bond cleavage is the key. So there are many different examples of regulated peptide bond cleavage like pro-hormone to hormone, and there are many different final enzymes that we generate, like precursor, it generates the mature insulin. Okay. And there is also peptide bond isomerization. This is intramolecular. SCR generates esters, cysteine generates thioesters, and these are different types of variations of peptide bonds are formed. Then there are new peptide bond formations. This is called transpeptidation. This is also interesting. This is called transpeptidation is a chemical reaction as a reversible condition of one peptide becomes another peptide through a protease, in which an amino acid residue or a peptide residue is transferred from one amino acid compound to another. So it is transferred from one amino compound to another. So this is called transpeptidation. So this is peptide deletion and or permutation plant lectin. So this is an example. So the protein formation changes, and this becomes from the peptide is transferred from one amino compound to another amino compound. So this is another type of like modifications involving peptide bond cleavage. Then there are modifications involving amino and carboxyl termini. As you know, the amino terminus, like the N terminus, there are many different variations that can be formed using the amino terminus, and these are the different N-methylation and N-glycosylation, and these are the different types of post-translational modification. Again, in the C terminus, that is the carboxyl terminus, you have the amide binding or O-methylation and N-alpha T by T modification. So all these are different types of C terminus modifications that take place. Again, we need not have to know everything, but maybe we can just keep in mind the examples and remember one or two examples for each category. Then again, the most importantly, the modifications involving the side chains, that is the individual amino acids. As you remember that the amino acids differ from one another based on these side chains, because you always have otherwise a central alpha carbon, you have a amino terminus and a carboxyl terminus. Okay, just like this. So you have a alpha carbon, this is called the heavy carbon, and this is one N terminus, this is the C terminus, and this is the side chain where we have all the differences. So based on the configuration of this R1 or R2, we have many different types of individual amino acids, and all these individual amino acids undergo many different types of modification. For example, in arginine, you have some different types of modifications, post-translational modification. In asparagine, you have N-glycosylation. This is a very popular type of modification, and this is often connected to, we are also working partially in this N-glycosylation modification in asparagine. This is connected to the disease of depression and stress. This is this takes place in neuroproteins. Okay, so asparagine. Then we have aspartate. There are several types of modifications. Then again, cysteine. Cysteine is a very interesting residue, and in cysteine you have many different types of modifications. For example, in S-palmutylation, S-palmutylation is also very well studied post-translational modification, which takes place in neuroproteins again for different types of diseases like stress-related disorders like Alzheimer's disease and neurodegenerative disorders in a generic term. So cysteines get modified, and they are often S-palmutulated, and they have different types of functionalities, which is not within our scope, but cysteine residues are modified or S-palmutulated. So like that, you have glutamate for different types of modifications, glutamine, proline. I'm just skipping this. You can just, if you're interested, you can go through and remember one or two in each category. And if you wish to discuss, if you wish to go into detail, you can always go into detail of each type of modifications. Now the question is how these modifications are made. How these modifications are made. Now we are saying, okay, the proteins are modified and these are the different types of modifications, but how these modifications are possible? So these modifications are possible either by non-enzymatic reaction or enzymatic reaction or something else. So what do you mean by non-enzymatic PTMs? Like non-enzymatic post-translation modifications are formed by spontaneous chemical reactions of amino acid side chains by diverse mechanisms, such as there may be some oxidations, like in the natural in the protein cytoplasm, or maybe in the nucleus. You can have certain natural spontaneous chemical reactions in the side chains, such as oxidation or glycation. And for example, protein glycation results from reaction between reducing sugars and amino groups of protein. Okay, so that reacts to protein glycation, and that adds to different types of PTMs. Okay, for example, slow oxidation modifies cysteine, histine, and methionine. Like deamidation results in ASN and GLN. Okay, so these are non-enzymatic natural chemical or spontaneous chemical reactions. Now there are some enzymatic reactions also, and this is most interesting, to be honest. And enzymatic reactions means some enzymes, like some other proteins, they mediate some specific types of post-translational modifications, and it's like you are having an external agent to change certain functionality of a protein permanently. For example, irreversible unidirectional reaction. That means there is no effect on the enzyme that is enforcing that, and it is irreversible. Like N-linked glycosylation, carboxyl methylation. So these are irreversible, unidirectional, permanent enzymatic reaction. So some other enzyme, they go and change another protein to add certain other molecules to it, to attach or to deattach. Some are reversible. Okay, so some enzymatic reactions may be bidirectional reactions, like signal amplification. So if it is attached, then some signal amplification is done. If it is deattached, then there is no more signal amplification, like phosphorylation. Okay, and dephosphorylation is also possible. So phosphorylation, as long as the phosphate is attached, and then again it can be dephosphorylated. The same protein can be dephosphorylated, and that enzyme, it is an enzymatic reaction, please remember. Then there are some reversible reactions. So like disulfide isomers coupled with protein folding processes, so they can form this and they can also reverse this. Okay, so this kind of reversible reactions are also possible, like the enzyme with the protein, they form something and then again they reverse back to the original state. Okay, these are called disulfide isomers. Now, with this, we complete the biological discussions of the post-translational modifications, and we have studied why post-translational modification, what are different post-translational modifications, how these post-translational modifications are done. So we have studied all these things in some details. This is not a complete discussion of all the detailed post-translational modifications that are available or known till date, but you have some idea that PTMs are absolutely critical and this is a hot research topic. So these days people are actually looking for prediction of post-translational modification sites in protein sequence. If you see a protein sequence, and you may need to know whether this particular cysteine residue is having a propensity to be modified in future. So whether this cysteine can actually be modified. Some can be modified, not all residues can be modified. So if you know that, then you can design certain experimental thing based on that. So you can actually have certain drugs designed with that and many other activities. So I'm discussing two publications where we are actually working on post-translational modifications in protein sequence. So there are two web servers that were designed. One is called AMS-3, and the newer version of this was AMS-4, automotive server. So you can actually submit a protein sequence there and you can actually get the predicted, you can select whether this sequence is susceptible to a specific type of post-translational modification, and you can always get back the result. So I'll discuss a bit in more details in AMS-3. In AMS-3, it is designed using artificial neural network. How you submit a query protein, and how you use the query protein, you actually the whole sequence. Suppose you have a 300 amino acid long sequence, and you can actually dissect it into overlapping short sequence fragments. Here the sequence fragments were dissected as 9 amino acid long sequences. So you have a central residue and 4 amino acid on the left and 4 amino acid on the right. So you have to predict the central residue, you have to check all the 9 residues. So in this paper, we actually identified 10 different physicochemical features. How these 10 physicochemical features were identified, that is also, and what are those physicochemical features that were also discussed in the paper. And it generates like 10 features for one residue. So 9 residues in all, so you have 90-dimensional feature space. And using all these 90-dimensional feature space, using the training data set and the test data set, you can actually train a neural network classifier and assess the efficiency of the classification routine. So what the efficiency was validated, efficiency was reported in terms of recall precisions and ROC, like receiver operating characteristics curve. And leave one out test, you know what is a leave one out test? That means if you have a small number of ground truth samples, often you actually need to do LOOCV test. That means if you have, say, 10 samples for which you know that this particular residue has been modified by that. Okay. For example, S palmutylation. So cysteines are L palmutylated. So if you have very limited number of samples, then that you know that okay, I have only 20 cysteines that I know were S palmutylated. So you have less number of training samples. Unless you train properly, it becomes difficult. So what you do, you train with 19 samples and leave one for test. Okay, you train with the 19 samples and try to predict the 20th sample. Okay. Now you repeat that for all the 20 samples. Like you take the first sample as test, the remaining 19 as training. Take the second sample as test, take all the remaining 19 as training. So this way you can have 20 different training models and you have all the 20 samples once considered for testing. So this model is applied quite often when you have very less number of training samples. So in this particular work, because of the gold standard training sample, gold standard benchmark datasets were, or ground truth datasets were less, so LOOCV method was used. So I can also show you the this is the paper, AMS-3 prediction of post translational modifications, and you can see the methods where you can see that different types of performance measures are given, and see, these are the feature selection process, and you can see that these are the features like information value for accessibility was considered. So you can see that scaled side chain hydrophobicity value was considered. Okay, so there are two different types of, say, hydration number is considered as feature. So side chain interaction parameter has been considered as features. So there are many different features that were considered, and you can see that there was a previous version of the server, and for this, like so for a specific type of PTM, so what is the recall, what is the precision, what is the AMS-3 with the new data set, what is the performance. So you can see that improvement is shown, and I can show you like the best PTM predictions. So they have, this is the best results in case of CDK and all these things. These are the best results reported. Likewise, I can, there is another version of this AMS server that is called AMS-4, where 80 different types of single amino acid post translational modifications are predicted, and each residue is represented using amino acid physiochemical features, and in this case, high quality index features were used. Like there is a database, you already know that this is called A index database. At that time, there were around 544 different types of physicochemical features present in the A index database. And what HQI clustering does, it identifies 8 very high quality features, then 24 high quality features, and it identifies actually 8 different clusters, and then there can be 3 different features in the clusters. When they are singletons, they have 8 different features. Then there is a 3 different features, mean 8 into 3, there are 24 HQI features, and there is a 40 HQI features if you have 5 per cluster. So it's a clustering approach, and it generates different types of different features. And then again multilevel perceptron is used, and then there is a consensus strategy is used using brainstorming technology, which combines multi-objective instances of machine learning algorithm and data fusion of different training object representation in order to boost the overall prediction accuracy. What is a consensus? Consensus gives you the idea, like when you have 5 different prediction results, then what is the best way of prediction? Like which classifier is the best? You can say, okay, I have 5 different classifiers, and I'll just take the classifier which gives you the best result. So that is just the best classifier. But it may so happen that there are certain complementary informations even present within some other classifiers, which the best classifier is unable to present. So often in machine learning, we take a consensus. That means, for example, the simplest consensus is a voting consensus. You have 5 different classifiers, and you just take a voting. And if you have a decision by 3 different classifiers, at least 3, then you can say, okay, then this is a true decision. Otherwise, I am not taking it as a true decision. Suppose you are saying that whether a test, in a test set, whether a cysteine is to be modified. You have 5 classifiers, and classifier is saying, okay, cysteine is going to be modified. Classifier 2 is saying cysteine is to be modified. Classifier 3 is saying no. Classifier 4 is saying again cysteine to be modified. And classifier 5 is saying no. So you have 3 yes and 2 no. Then in a simple voting, you say, okay, then I am saying that cysteine is going to be modified. So this is a simple brainstorming or voting consensus. But here we are saying that we have a variable consensus scheme. Variable consensus scheme means if you have, in this case, you have 5 different classifiers, and if you say that, okay, I have a cysteine, and only classifier 1 is saying that it can be modified. Classifier 2 saying no, 3 no, 4 no, 5 no. So you have only one classifier saying cysteine is to be modified. That means the quality, instead of voting, we are stressing on the quality. That means this is a 1-star quality. We call it N-star consensus. Here it is a 5-star consensus. Here only one classifier is suggesting the cysteine to be modified. That means its quality is 1-star quality. Then, if 3 classifiers say, okay, the cysteine to be classified, then it is a 3-star quality. If all the classifiers somehow say that no, this cysteine is modified, then we call it as a 5-star quality consensus. And you know that result is good, because in the other case we were just considering the voting. Okay, if 3 classifiers say that the result is positive, and if all the 5 say the result is positive, there is no difference in the result, because in the voting they win. The majority wins. But what is the strength of the majority? It is just slightly above the midway mark, or it is overwhelmingly consensus. It's a full consensus. So that you can actually tune, and you can have a quality of the consensus result. So here in this work, in AMS-4, the quality consensus was implemented for 88 different types of proteins, and you can see that if you give an input as a FASTA sequence and sequence analysis, it is formed into say 9 amino acid long overlapping sequences, sequence 1, sequence 2, sequence 3. Then you have different databases and they have 88 different PTM types. And what you have different feature extraction, like we use 10 in AMS-3. I said like there are 10 AMS-3 features, and there are high quality index features, like there are 8 high quality index features, there can be 24 high quality index features, also 40 high quality index features. Then what we did, we actually trained based on backpropagation learning mechanism. Okay, and what we can do, we can have, for example, we trained on AUC optimized network, then recall optimized networks, and precision optimized networks. So, for example, if we have three consensus, that means AUC, recall, and precision optimized network, then we can apply a 3-star consensus mechanism. If we have 10 different features, and we can have a 10-star consensus mechanism, that if all the different networks are giving it as a positive, then we call it as a positive, and the quality can be varied. See here, if we go through this particular diagram, it will be more clear. For example, for example, HQI8 feature set, we have 10 different A-nets prepared. How 10 different A-nets? Because by varying the architecture of the neural network, like varying the hidden neurons of the neural network, you can generate 10 different A-nets. From that, you can get the best A-net. Okay, so you can get the best R-net with the best architecture of the neural network. So using this, you can have a C3N. That means you have the best C3N classifier. That means for best A, R, or P network. Likewise, for the 10 A-nets, you can have a 10 consensus classifier, which is a AUC optimized. Again, from R-nets, you have a recall optimized C10 classifier. AUC and R combined, you can have C20 classifier, C20 consensus, and all 30, you can have C30 consensus. Again, if you consider the best nets, then you have a C3N for the HQI8. So if you consider only HQI8 feature set, you can have a 3 consensus, you can have different 10 consensus, you can have 20 consensus, and finally you can also have a 30 consensus result. That means considering say for example a 30 consensus or C30N network, that means we have 30 different classifiers predicting whether a cysteine is true or not, cysteine is to be modified or not. And if the cysteine, if one classifier only says that cysteine is to be modified and all the 29 other classifiers say cysteine will not be modified, then the result will be considered as 1 star. The result is all still valid. And if we assess the overall system performance based on 1 star result, where at least one classifier predicts it as positive, then it is called a 1 star consensus. If we take a 30-star consensus, that means all the 30 classifiers have to say that this is a this will be modified. So sometimes we'll see that the best results in terms of recall, considering the type 1 and type 2 errors, are often found with 1-star or 2-star consensus. That means at least one classifier says that residue is positive, then it gives the best result. But when we discuss with the biologists, they often say that they are often interested with the higher star results. That means there are less chance of false positive. Okay, like if you consider a 30-star consensus in a 30 consensus situation, that means those values or those positives that you get are probably genuinely positive, and there are less chances to have a false positive information there. So that is why consensus prediction is always important, and this work is actually widely cited because of this consensus prediction. So this is all about like how you can actually use machine learning algorithms for prediction of post-translational modification sites. You'll have to very carefully read both AMS-3 paper as well as the AMS-4 paper that is supplied with this, and you need to understand how these classification mechanisms work, especially in case of AMS-4, because AMS-4 by and large is a superset, and AMS-3 work was mostly improved by AMS-4. So if you study AMS-4, more or less you will understand the AMS-3 work. But if you want to start and understand the general classification mechanism, probably AMS-3 will be a bit easier for you to understand because it uses flat classification mechanisms with 10 different features. So it will be a bit easier to understand, and in AMS-4 follow carefully the consensus mechanism. With this, we end this today's lecture, and we have studied in detail like what do you mean by post-translational modifications, its purpose, biological significance, and what are the different types of post-translational modifications, and ending with like the computational mechanisms like how you can predict post-translational modifications. Thank you very much. Thank you for your question.